In this video, we're going to look at goal setting plant style. In other words, what's the point of being a plant? Now, if it was day one of the term for plants at plant school, they would have to do goal setting. And what would you actually set as a goal if you were a plant? Well, obviously, you would want to be the best at photosynthesis. So let's have a think about photosynthesis. Remember, all plants are photoautotrophs, so they use light, photo, to self-autotroph, feed. So they use light to feed themselves. So in our word equation for photosynthesis, plants take the gas, carbon dioxide, and some water, and capturing the sunlight's energy using a molecule called chlorophyll, they can combine the water and the carbon dioxide together to make a carbohydrate called glucose and oxygen, a gas. Now that oxygen is used by the plant for respiration, but a lot of it is an unwanted byproduct, and so it gets released into the atmosphere for other things to respire. And the glucose itself can then be used to make lots of other products, like other carbohydrates, like starch. It can have its atoms rearranged to make fats or lipids, like sunflower oil or coconut oil. It can also add in extra atoms to make amino acids for proteins or nucleic acids for DNA. So basically, this is the starting point for all the organic building blocks of plants and therefore everything that feeds on plants. So when we are thinking as a plant, setting goals, we want all the structures of the plant to be maximizing the photosynthesis potential of that plant. So let's start at the bottom with the roots. Now, the point of the roots is to anchor the plant, to keep it in one place with its leaves up in the air, capturing sunlight and being exposed to carbon dioxide. The roots also absorb water. So we've got anchoring the plant and absorbing water. And whenever water moves across a cell membrane, that is moving by osmosis. And of course, we just saw the photosynthesis equation. Water is a raw material for photosynthesis. But we also absorb mineral salts. And because those mineral salts, these are things like iron, which might be needed for some enzymes, or magnesium ions, which might be needed to make more chlorophyll molecules if the plant is growing. These need to come into the plant by active transport. So we've heard about active transport before. This is the movement of things from a low concentration up their concentration gradient to where it's more highly concentrated and energy is needed for that to happen. So we've anchored the plant, we've got water in by osmosis, we've got mineral salts in, and also we can transport the water and the mineral salts through a structure called the xylem. So when we look at root structure under a microscope, you see all of the transporting tissue is concentrated in the very center of the root. This makes it incredibly sturdy and upright for its ability to hold that plant upright and anchor it well. The xylem is forming a cross in the center, and that's very sturdy tissue. It's actually dead tissue. It's kind of like hollow straw at the center. And the rest of the root really is just packing tissue. So all of this is called parenchyma. It's just sort of packing tissue where the cells just push against each other, filled with water, nice and turgid. And then there's an epidermis around the outside, which is nice and tough. So that's a root structure there. So when you have your roots, they are supporting the stem. The stems hold the leaves right up into the sunshine, supporting the plant. There's also vascular tissue in there, transporting water and mineral salts in the xylem, like it was in the roots. And there's another transport tissue called phloem, which is moving around all those photosynthetic products like sugars and amino acids, anything else that's dissolved in the water. When you look at a stem under a microscope, this is a slightly different structure. No longer is the vascular tissue, the xylem and the phloem, concentrated in the middle. It's now around the outside. So we've got some flexibility of movement. So there's our vascular bundle there. There's another one next to it. And you can see they're arranged as a circle all the way around the stem. And we've got the phloem, on the outside, so that's our phloem, and the xylem is on the inside. In this case, it's being stained red. And again, the rest of the tissue is really just packing tissue, just cells that are full of water pressing against each other, holding that stem structure upright. So you can hear from what I'm saying that if you've got a plant that's lacking in water, these packing tissue cells will not be able to press so tightly against each other, and the stem would start to bend and flop over. So let's have a look at what happens as the stem becomes the leaves. So obviously the leaves are our factory, the site of photosynthesis. In order to maximize their effectiveness, we want them to catch the maximum amount of light and to have diffusion happening really quickly. So we want the minimum amount of diffusion distance for that carbon dioxide to get in to the photosynthesizing cells. So we want large, thin, flat leaves, and when you look up through the canopy, you will notice that they form a mosaic pattern. They try not to block the sunlight from the leaves below. So they still have those transport tissues to move things around, and when you look at the cross section of the leaf, we've got a vein there and another vein here. So we've got xylem on the top and we've got phloem underneath. On our vascular tissue or our veins. You can see the veins when you look up through here into the sunlight. So you can see the veins there. So there's lots of transport tissue to bring the water and take away the products. Obviously, leaves are green. Without chlorophyll, they can't trap the sunlight. They actually pick out the blue and the red wavelengths of sunlight and trap those and capture that energy and put it into those organic molecules. And strangely enough, leaves have holes in them called stoma or stomata, and they are for the gas exchange, and they're down here. You can see one there and you can see one there, and we'll talk about those in just a minute. So if we have a slightly closer look at our leaf in cross section, so this is a sort of diagrammatic view of those cells like you would look under the microscope. We can see that the leaf is made up of many different layers of tissue. Starting at the top, we have cells in the upper epidermis. These guys actually make waxy cuticle, which is see-through. It's a protective layer that stops evaporation of water to some extent, and the thicker that layer, the better they are at preventing evaporation straight out through the hot surface at the top of the leaf. But these cells don't have any chloroplasts in, so their job is not photosynthesis, it's just to make the waxy cuticle. Here are our photosynthesizing cells. So the chloroplasts are found within our palisade cells, and these are standing tall and like soldiers, really closely packed, and you'll notice in them that there are lots and lots and lots of chloroplasts. So all around here we've got green chloroplasts in all these cells. There are still chloroplasts a little bit further down, so you can see some chloroplasts down here, but they become less frequent in the cells down here, in the spongy cells or the spongy mesophyll layer here, because there's hopefully less sunlight getting through. So the sunlight has come in up the top here, straight through these colourless cells, and then it gets trapped in all of these chloroplasts. Maybe some of it comes through down here and gets picked up there, but most of the sunlight is getting trapped in these top layers, in the palisade layer. Right down the bottom, we have another clear layer of lower epidermis cells, again making a waxy cuticle there. And you'll notice our guard cells on either side of the hole where our gases come in and out, our stoma down the bottom. There's the vascular tissue, the xylem and the phloem are vein in the leaf, transporting materials to and from the leaf. Let's have a final closer look at the stoma. The very bottom, right on the underside of the leaf, is the most common place to see the stoma or stomata, and you will see on the underside of the leaf a structure like this. That black little ellipse shape in the center there, that's actually the hole, the stoma. So that's this bit here. And on either side, you've got two cells that look a little bit like sort of sausages, and they are called guard cells. So there's our guard cell there, and they control whether the hole is open or closed. When it's open, which is usually when it's sunny. Anything that has evaporated into the air holes in the spongy layer can leave, as you can see from this big blue arrow, water vapor can leave through the hole. This is really important. It drives the movement of water in the xylem, but really we want the stoma, in terms of goal setting for these plants, to be allowing the carbon dioxide into the cells. Because the carbon dioxide needs to diffuse and reach not just this first layer, but all the way up to the top of the leaf, where the palisade layer cells are that are doing the maximum amount of photosynthesis. So that's what a leaf would want to do if it was goal setting. It would want to maximize its ability to capture light, to bring water up from the roots by osmosis up the xylem into the leaf. It would want to be able to get rid of its raw material, its products, so through the phloem, as well as get rid of oxygen that it's made, and it needs to be able to take in carbon dioxide. So anything that it can do to maximize any of those processes would be a great goal for this leaf, for these plants to set.